the two types of protein categories that you kicked off your explanation with were biocatalysts and antibodies. So it sounds like you're starting to get towards that these are two areas that Exazyme specializes in. And so you have two different sets of clients, I guess.
Yeah.
One of which is interested in these kinds of industrial applications like BASF. And then the other set is this medical application, cancer detection.
Actually, yes. So biocatalysts are interesting to chemical companies and industrial biotech companies. The medical application, let's say pharma is interested in both antibodies and biocatalysts because they use biocatalysts in order to make even their normal drugs in a more sustainable and profitable fashion.
Nice. Yeah, that makes a lot of sense. So, yeah, maybe you can go into, I don't know, I don't know to what extent, if there's, without obviously going into proprietary secrets that you can't disclose on air, are there some concrete examples you can give in each of those sectors?
Yes. So we've up to now mostly worked on biocatalysts. So one of our recent success cases was where we engineered a biocatalyst that plants use to fixate CO2. So basically take out the carbon atom of CO2 and use it to build up biomass. That's what plants do all day long. They take out CO2 from the air and use the carbon atom to build out sugars and everything else basically that they make using the energy they get from the sun. And our collaborators designed a new biocatalyst that can do this. Their goal basically is to enhance plants to take up more CO2 and store more CO2 than they usually do. And they were trying standard methods for optimizing this biocatalyst, and they made a lot of protein variants. The first method they used was what in machine learning we call student descent. So somebody reads papers and thinks very deeply about what can I do, and then they try out stuff. That's what they did first. Then they came along with the brute force method, which is basically an evolutionary algorithm implemented in the wet lab, meaning you just create random changes to the protein sequence, random mutations, that's where it's coming from, and then screen for which variant is improving. And this way they generated over 15,000 different variants in the lab. And they got to a certain point, and that was kind of it. And we started talking to them, and they were very cautious and said, yeah, okay, so this AI thing, everybody's talking about it, so yes, okay, we are willing to give it a try. We gave them 10 more suggestions after the 15K that they made and measured previously. And of these 10 suggestions, 2 improved the performance. 1 cut the energy demand in half, and the other tripled the catalytic speed. So yeah, this is our last success case, big success case.
Yeah, that's wild. So can you dig into a bit the AI, the machine learning that you're using to make those recommendations? I mean, that's wild. And they must have been, were they blown away?
They were very surprised. They were quite blown away, didn't imagine why the hell this worked, because like the PhD student who worked with us, he said, yeah, but it's very far from the active site. The active site is kind of the place where the action happens in the biocatalyst. So the mutation your AI suggested is very far from the active site. Do you have any idea why it suggested you there? And I was like, no idea. It's a black box even to me.
Yeah.
And so they were trying to get the 3D structure of this protein in order to understand it. Yeah, and maybe this is also an interesting part because many people know AlphaFold. People in biocatalysis use AlphaFold because it gives them the 3D structure that a protein sequence translates to. Mm-hmm. It's called folding. And from the 3D structure, they are trying to gain insight. We take a shortcut. We don't go through 3D structure, through folding. We directly go from the sequence to the performance that you're looking to improve, basically with a regression model. And the methodology we used, I won't go into detail, but really the first method that we used is, of all things, kernel methods, which many people still might connect to support vector machines. It didn't change for quite a long time because there was no need to go to another method. Of course, we also use deep learning as well nowadays, but we're really big believers in metrics. So whatever performs best, that's what we use.
Yeah, and we're going to talk in more detail later on. We have a whole topic area around how deep learning is not always the answer and shallow methods can be more useful.
Yeah.
So it's not surprising for me to hear you say that at this time. And yeah, we'll dig into that more later. For now, you said that you used a kernel method. And yeah, for me, when I hear kernel method, I think only of support vector machines. And so, but the way that you phrased it there, it sounds like it's something else.
Yes, so support vector machines are really old-fashioned now in kernel methods.
Oh.
Still performing very well, but old-fashioned in the sense of they've been around for a long, long, long time. And the way people thought about kernels back then has gotten several updates. And it's by far not as fast as deep learning research, I guess mainly because people in kernel methods, they insist on theory. And I think it's a very nice property of many shallow methods, I guess, that you can do theory. But the fact that you can tinker so much and so easily in deep learning is probably what has gotten deep learning this big following.
Yeah, deep learning famously, there's very little explanation for many of even the core capabilities of these models. It's just, it works.
Yeah, true.
Okay, well, that's really cool. And so maybe it is something proprietary, and maybe it's something you can't disclose, but are you able to give us, if it's a kernel method that's not an SVM, are you able to tell us kind of generally what this method is called? Or
it's not published, actually.
Okay.
So that's why I can't give a name, because I never had to find one for any paper.
Okay.
But it's basically, well, unsurprisingly, it's a way to embed a sequence into an embedding space. This is very standard, right? So in deep learning, you would, for example, embed into a fixed dimensional space and then do a regression problem from that. Or maybe you would use the regression variant of transformers with an artificial token in the beginning, whatever. Here, yes. So it's an embedding into an infinite dimensional space, in theory at least. And then you do a regression problem from there, which works with very few data points. That's the big advantage of this.